The Cell Cycle Regulation in the Cell by Russell Carnes at Grand Canyon University. We have four phases of the cell cycle. First, we have the G1 stage, which is where our cell will increase in size through cellular components that are duplicated. Then we have our S phase. Our S phase is our DNA synthesis. DNA replication, where as much as 46 chromosomes would have 23 pairs. This is replicated by the cell. Then we have our G2 phase. Our G2 phase is our growth and preparation for mitosis. This is where our cell grows more and our organelles and proteins develop in preparation for the cell division. Then we are followed by our M phase. Our M phase is our mitosis phase. Mitosis is followed by cytokinesis, which is our cell separation. The formation of these two form identical daughter cells. Our cell cycle. Our cell cycle is the most basic function is to duplicate the amount of DNA. Replicating the DNA is a very fragile process. Broken chromosomes are often the source of this DNA arrangements that can change the genetic program of a cell. These cells then can trigger the growth advantage in a single cell of your body. And when that cell continues to divide, this is where tumors could arise. Unfortunately, our cells have a defense mechanism that will also help us shield us from these damaging events that will occur. The cell cycle control system. The cell cycle control system basically has G1 checkpoints, a G2 checkpoint, and an M checkpoint set in it. The cell cycle control system has been compared to a kind of a control device, kind of like an automatic washing machine. Like the washer's timing device, the cell cycle control system produces its own driven but built-in clock. However, just like a washer will do, it is subject to both internal and external control, such as a sensor that detects when the tub may be filled with water, or have an external adjustment, maybe such as activating the start mechanism. The cell cycle is regulated at certain checkpoints, just like the washing machine, by both internal and external controls. A checkpoint in the cell cycle is a critical control point where stop and go ahead signals can regulate to the cell cycle. The signals are transmitted within the cell by the kinds of signal transduction type of pathways. Cyclin is one of these main pathways, and its CDKs, which is cyclin-dependent kinesis, are the principal regulators of the cell cycle. This progression through the activation of the cell cycle are these checkpoints that are set up. These are the core of the complex that composes the mechanism which drives the irreversible transitions that must be successfully negotiated for the cell in order for it to successfully navigate the cell cycle without proliferation. The cell cycle control do not apply, such as cancer cells. This means that cancer cells do not respond normally to the body's control mechanism. They stop at the normal checkpoints. If and when they stop dividing, they do so by abnormal points eternally. Cancer cells may not need to grow and have factors that make them grow and divide. They may make their own growth factors. They may have an abnormal cell cycle control system. Thus, loss of the cell cycle control leads to these cancerous cells. Interphase, or what I like to call is the G1 phase. This is where we have recovery from previous cell divisions. Cells doubles, it organelles, it grows in size, and then it accumulates raw materials such as DNA synthesis or DNA replication. The G1 phase is often referred to as the growth phase, but because of this time in which the cell grows, during this phase, the cell synthesizes using various enzymes and nutrients that are needed later on for its DNA replication and cell division. The duration of the G1 phase is variable, and it often depends on the nutrients that are available in the cell. The G1 phase is also when cells produce the most proteins. During G1 phase, a cell will grow in size, and then it will start synthesizing mRNA. With this, then proteins that are required for the DNA synthesis are required. Once this requirement of proteins and growth are complete, the cell then will enter its next phase of the cell cycle, which is the S phase. The duration of each of these phases, which includes the G1 phase, is different in many types of cells, depending on what type of cells we are talking about. In the human somatic cells, the cell cycle lasts generally around 18 hours. And in the G1 phase, it can take up to 1/3 of that time. However, in a lot of embryos, such as sea urchins or Drosophila, the G1 phase is barely even noticed and has a very defined gap. If one exists between the end of mitosis and the S phase 2, G1 phase then is synthesized. Factors such as nutrient supply, temperature, room growth, these are all very sufficient. Some sufficient nucleotides such as maybe amino acids may be present in order to synthesize also with mRNA or with other proteins. These types of temperatures make it optimal for cell growth. In humans, the normal temperature for good cell growth is around 37 degrees Celsius. Let's talk about the safeguards of the cell cycle. The safeguards of this cell cycle that are set up subphases like G1 phase, what happens during interphase. The GO phase, which is often referred to as the G0 phase, or the resting phase, is a period in the cell cycle in which cells exist in a type of state. GO phase is viewed as either an extended G1 phase, where our cells are neither dividing nor preparing to divide. These have distinct stages that occurs outside the cell cycle. Some types of cells, such as nerve and heart muscles, can become this way when they reach maturity. As our figure shows, each place what it's going through. First, we have our mitosis, then our cytokinesis, which is followed by our G1 phase, where the cell will increase in size, its structures are duplicated, and the mitochondria and chloroplast may divide through binary fusion. Then we'll have our S phase. The S phase is duplicated. Each chromosome there will then have its two sister chromatids, which was followed finally by the G2 phase, where DNA replication is checked for maybe errors or incorrect repairs. Now, although G1 is a phase of the cell cycle, it has 4 subphases that also divide it and describe its process and functions. The 4 stages are very complex. They have progression and assembly that are required. Competitiveness refers to the process by which a cell absorbs its nutrients and its things from outside of the cell membrane. All right. In turn, these materials will then enter the cell in the entry of the subphase. They are used to help then the cell grow, and which takes place during its progressive subphase. The assembly of the subphase refers to a process by which all of the materials then can come together in the cell to complete the G1 process at this restriction point of stages. The growth of all cells is governed by the cell cycle, including cell division. Before a cell can divide, many processes then must occur, including the proper duplication of the chromosomes. Once that has occurred, the cell cycle ensures that all of these processes can occur normally. Otherwise, the cell will then cease to progress, and it also will die. The S phase. The S phase is where an enzyme called heliocase will unwind the DNA strand in the same way that sometimes that you would unzip a zipper. The S phase of the cell cycle occurs during interphase, before mitosis or meiosis, and is also responsible for the synthesis or replication of our DNA. In the same way, the genetic material of the cell is doubled before it enters mitosis or meiosis, allowing there to be enough DNA to be split into two daughter cells. The S phase only begins when the cell has passed the G1 checkpoint and has grown enough to contain double the DNA. The S phase is generally halted by a protein called the P16. The P16 is a protein that basically halts this S phase when it wants to happen. The most important event, though, that occurs in the S phase is definitely the replication of the DNA. The aim of this process is to produce double the amount of DNA. By doubling the amount of DNA, this provides the basis for a chromosome, which sets up the daughter cells. DNA replication then begins at these points where regulatory pre-replication complexes are then attached to the DNA in the G1 phase. These complexes can then act as a signal by sending this signal for where the DNA replication should start. They are removed, though, in this S phase before replication begins so that the DNA replication doesn't occur generally more than once. This is a safeguard. Then we have an enzyme called DNA polymerase. Then binds nucleotides to each of the single templates. The DNA strands using the complementary base pairs rule, which generally means C to G and T to A, which is for our different bases, and these will code in certain ways. T will bind with A, C will bind with G, and so on. This enzyme, which is called DNA polymerase, is basically in a single strand or template of DNA. This DNA strand, using this complementary base pairing rule, adenine to thymine and cytosine to guanine. In this way, a new double DNA helix can be created and formed that is identical to its original strand. Among the array of proteins now that can be used in this process, we can have sliding clamps, we can have clamp loaders, we can have clamps that zip or unzip the strand that even work with some binding proteins. We have new fragments that are in place. We have proteins such as polymerase, and prisomes that basically bind things that are shown in this figure. Among the array of proteins at the replication fork, where we're going to basically zip or unzip is DNA polymerase. It is often central process in this replication. These important enzymes can only be added with a new nucleotide, triphosphates, onto the existing piece of DNA or RNA. Now, once we've gotten this established, they cannot synthesize DNA from scratch. From a given template, what they do is another class of proteins will then fill its functional gap. Unlike DNA polymerase, RNA polymerase can synthesize RNA as long as DNA template is available. This particular feature of this synthesis, or de novo synthesis, is similar to what happens during our messenger RNA transcription. Eukaryotic cells, they basically possess an enzyme complex that has an RNA polymerase activity, but works in DNA replication. This unique enzyme complex is called DNA primase. Interesting enough, this primase generates small to a 10 nucleotide long RNA primers from a DNA template. Basically, the red portion of this Okazaki fragment, the RNA primers produced are later replaced by DNA, so that the newly synthesized lagging strands is not a mixture of both DNA and RNA, but consists exclusively of DNA. This is the main point. The chemical properties of DNA and RNA are quite different, and DNA is preferred. It has storage material for the genetic information of all cellular organisms. So now it can have a reinstallment plan. This reinstallment of the DNA strand is very important. In prokaryotic cells, DNA primase is its own entity and works in a complex with the DNA heliocase. However, in eukaryotic cells, DNA primase is associated with another, which is polymerase. DNA polymerase, which basically initiates the leading strand in these fragments. At present, we have no evidence that DNA primase binds to DNA heliocase in eukaryotic cells, but it is also likely that some connector proteins must coordinate DNA and unwind. DNA synthesis then would have to be initiated in eukaryotic cells. All right, our G2 phase. Our G2 phase is called the gap phase because no further cell division or specific process takes place. Instead, there are higher levels of preparation and checkpoints to make sure everything is in place for the successful mitosis. The G2 we'll start with. The G2 is called a gap phase because no further cell division or specific processes or progress can take place. Instead, there are higher levels of preparation and checkpoints to make sure everything is in place for successful mitosis. Before the G2 phase can start, each chromosome of the cell must have been duplicated by then, and the process is required for extracellular membranes and cell structure must be present at this point. At the beginning of the G2 phase, the organelles such as the mitochondria and the lysosomes will then start multiplying. These organelles have their own DNA, and they can start dividing independently. But the cell itself has to create extra ribosomes to satisfy these needs of the basically two new daughter cells. As you can see in our figure, we have our chromatin threads, our nucleus, and our nucleolus that are inside. As the cell is growing, multiplication will start taking place. our G2 phase. Our G2 phase, this is the last part of our interphase in the cell cycle. The DNA has been replicated at this point and is ready to divide and prepare for mitosis or meiosis. As you can tell from our organelles are then produced and will increase our volume of our cytoplasm. Our figure here will show our mitotic phase, or what I like to call PMAT, prophase, metaphase, anaphase, and telophase. Then we'll have our first gap G1, we have synthesis, and our second gap G2. This is the last part here of interphase of the cell cycle. The DNA has been replicated and it is ready to divide and prepare for mitosis or meiosis. Now we'll begin our final step of interphase. In the final step of interphase is the second gap phase, or what I like to call the G2 phase. During this step, the cell makes everything it needs in order for it to divide. As we can see here, even more the cell grows, the volume of the cytoplasm will also increase as well. Now we'll also have new proteins that are formed. These new proteins and organelles are made that will be really needed during our nuclear division. An example of this could be microtubules. They basically help separate the chromosomes during the cell division, or what we were on in our M phase, but are created also during G2. G2 is also the shortest phase of interphase. It is when these organelles and proteins that are necessary for cell division are produced. The cell basically requires a bunch of proteins then and other stuff in order to separate these chromosomes and divide the cell in half. All of these materials then are produced during our G2 phase. We can also have the cell that will have checks in it. We can have checks for DNA damage that can stop during this phase. Continuing to replicate the cell, if we see that DNA is damaged, we'll basically cause a bunch of damaged cells. So we have this checkpoint. The checkpoint is in the S phase, or called the G2/M checkpoint. This checkpoint will then stop cell division immediately. By stopping cell division, if the cell is damaged, this checkpoint then can get rid of the cell. These are very important checkpoints that prevent not only uncontrolled growth, but can also cause cancer cells that can later multiply and cause difficulty. Now we're going into our M phase. Here our figure shows how our microtubules have basically migrated to both ends of our cell and have our long tubes that are basically reaching in towards the, generally pointed in towards the equator of the cell. Then we'll have progression through the M phase where they're pulled apart, pulled tightly, and we'll definitely have long microtubules then that will form to have basically a contractile ring. It's mostly actin and myosin filaments, and the cell will start to pinch. After the chromosomes now have condensed, there are two distinct cytoskeletal machines that are assembled in this sequence to perform a mechanical process of mitosis and cytokinesis. Both machines then are ready to disassemble rapidly after they have completed their task. To produce two genetically identical daughter cells, the cell has to then separate its replicated chromosomes and allocate one copy each of a daughter cell. In all eukaryotic cells, this task is performed during mitosis by a bipolar mitotic spindle. This mitotic symbol can even be seen in slides, which is composed of microtubules and various proteins that interact with them. We also have that is included a microtubule dependent motor protein that can be found. Now there are different cytoskeletal structures. These different cytoskeletal structures are responsible for cytokinesis. In animal cells and many other eukaryotic cells, it is the contractile ring, which can be the most predominant. In most plant cells, it is the phagoplast. The contractile ring contains both actin and myosin filaments and forms around the equator of the cell, just under the plasma membrane. As the ring contracts, it pulls the membrane inward, therefore pulling the part and dividing into two cells. Now with plant cells, which have a cell wall to contend with, they divide their cytoplasma by a very different mechanism. As we discussed later on, instead of using a contractile process that acts on the plasma membrane, these construct new cell walls and form within that new cell between the two sets of the replicated chromosomes. Our cell cycle progression here then shows starting with the G1 phase to the S phase, to the G2 to the M. We also show how transcription is then therefore applied. These complexes basically activate specific steps in the cell cycle by degrading basically a cell cycle proteins such as cytokines and CDK inhibitors proteins. Thank you for our presentation today on the cell cycle.